First of all, I'd like to thank the Brick webinar for inviting me out to give this presentation today. My name is Brian Lee. Today I'll be talking about a paper that was recently published in eBiomedicine this year called High Dimensional Profiling of Regulatory T cells in Psoriasis Reveals an Impaired Skin Trafficking Property. And so just to briefly introduce psoriasis, psoriasis is a chronic T cell mediated inflammatory skin disease that typically affects around 2% of the global population. It's characterized by thickening and scaling of the epidermis due to hyperskin cell keratinocyte proliferation. And there are various types of psoriasis, with plaque psoriasis being the most common, with around 80 to 90% of people having plaque psoriasis. The most common symptoms usually are red plaques of skin, covered with silvery scales, dry skin, and itching. And so going into the actual mechanism of pathogenesis of psoriasis, it normally starts off with a potential environmental trigger, such as trauma, stress, or pathogens. That trigger causes the keratinocyte to be stressed, and releasing self-nucleotides and antimicrobial peptides, which leads to the activation of PDCs. These PDCs then release interferon, TNF alpha, IL-1 beta, IL-6, activate MDCs. These DCs then release these pro-inflammatory cytokines such as IL-23, TNF, and IL-12. And one of the main players in pathogenesis for psoriasis are Th17 cells. And so these Th17 cells will then go on to release IL-17, IL-22, which further activate the keratinocytes into proliferating more, and then releasing more TNF alpha, IL-1 beta, IL-6, going to this feedback loop cycle of continued proliferation, continued inflammatory process. And so many therapies currently involve treating this Th17 axis by using an IL-17 or IL-23 inhibitor. And there's also kind of other therapies that also target beforehand with TNF alpha inhibitors. But yeah, this is the main kind of mechanism of pathogenesis. And particularly of importance are Tregs. And Tregs play a key role in regulating and suppressing the immune response, and by inducing immune homeostasis and self-tolerance. And so Tregs can function in multiple mechanisms to inhibit other T cell responses. They can inhibit T cell response by IL-2 consumption, by releasing of suppressive cytokines such as IL-10, TGF-beta. They have inhibitory receptors such as PL1, TLA4. They can also directly kill other effector T cells by granzymes. And so for a long time, the role of Tregs in pathogenesis of psoriasis has been proposed in several previous studies, but the actual mechanistic identification of these Tregs, their exact function, and their exact properties have yet to be elucidated. And so normally Tregs are quite important in controlling this Th17 imbalance. And so many people have thought that an imbalance of Tregs, Th17 cells, may be another key reason as to why psoriasis continues on in development. And so in previous studies in terms of Treg functionality, recent studies have shown that psoriatic Tregs are functionally impaired compared to healthy Tregs. This can potentially be due to the pro-inflammatory milieu, such as IL-6 in the skin, which downregulates Treg activity. There's also the presence of inflammatory cytokines, such as IL-1 beta, IL-6, IL-21, and IL-23, in addition to TGF-beta that can skew Tregs towards Th17-like phenotype due to Tregs having a plastic effect. In terms of actual Treg homing, around 60% of circulating Tregs will express cutaneous lymphocyte antigen, or CLA, and this is an essential skin homing receptor involved in the migration of Tregs into the skin. And so the goal of our project was to characterize these skin homing Tregs in psoriasis patients and compare them to healthy donors to see if we can gain some insights into their actual mechanism and their phenotype. And so we employed mass cytometry, otherwise known as CyTOF, or cytometry by time of flight. with this unique capability of being able to do high-dimensional characterization of over 40 markers at the single-cell level using metal-conjugated antibodies with minimal signal overlap. And so compared to flow cytometry, normally where you have a lot of signal overlap due to the wide range of fluorescence, in CyTOF, the mass overlap is quite distinct. So there's very little overlap between markers, allowing for this kind of high-dimensional profiling of over 40 markers. And so we designed a CyTOF panel mostly to profile Tregs in depth, but also aiming to profile Tregs based on their chemotactic profiles. And so the way we designed the study is we took 32 psoriasis patient samples with 32 age-sex matched healthy donor samples. We then banked those all together, and then we thawed all of those samples for each batch. We then labeled those cells with RH103 to be able to identify dead cells. We then stained them with a pre-aliquoted frozen antibody cocktail with these markers shown here. Then barcoded all of those samples together into a single tube for batch acquisition on this CyTOF Helios mass spec. And this was done in collaboration with our with Mount Sinai, with the Human Immune Monitoring Center at Mount Sinai. And so once we got back data, in terms of the actual annotation of these cellular subsets for downstream analyses, there are a few ways that one can approach this analysis. Method one historically has been the most common approach, and this is by manual gating using FCS software such as Flojo or Cytobank. But the issue with this is that oftentimes the gates can be very subjective, and they can be quite time-consuming to gate every single sample and every single population within each sample. Another method would be using unbiased clustering, using clustering algorithms such as Phenograph or FlowSum. And normally it's quite easy to apply these approaches for clustering high-dimensional data. However, they can significantly be affected by batch effects, and the unbiased clusters that come out of these algorithms are often hard to interpret and often do not adequately capture known biological relevant subsets. And so instead of these two approaches, I developed an in-house custom semi-supervised annotation pipeline, where essentially from each sample, we downsample each of the samples using k-means clustering. And so we intentionally overcluster each sample into 500 k-means clusters. We then aggregate all of those k-means clusters across all of the samples into one kind of meta cluster. And then we use a heat map-based annotation to annotate these k-means clusters in a tiered fashion, starting off at a very broad level with just T cells, B cells, NK cells, monocytes, and so on. And then from each of those subsets, we further subclustered using metaclustering, and then in particular, trying to define Tregs. And so after that annotation process, so this is the kind of broad immune landscape of psoriasis versus healthy donor PBMCs. So we can see here, this is a UMAP of the healthy controls and a UMAP of the psoriasis samples, along with their marker annotation. And when we compared the frequencies of the cell types between psoriasis and healthy controls, we found that Tregs, so CD4 Tregs, are quite elevated in psoriasis compared to the healthy controls. And we also found that monocyte subsets were significantly reduced in psoriasis compared to healthy controls. And so because of this phenomena, we decided to further profile the CD4 Tregs. And so what we did is we did unbiased metaclustering of those Tregs using the Leiden clustering algorithm. And what we can see here, this is a UMAP of those Tregs colored by the Leiden clusters. And what we can first appreciate is that in psoriasis, there's actually a marked reduction in the frequency of CD4 Tregs, of these CLA high CD4 Tregs. And as I mentioned before, CLA plays a very important role in homing of Tregs into the skin. And what we found is that not only is the frequency of these CLA high Tregs reduced, but also the marker expression. So this is a UMAP of CLA expression on our Tregs, and you can see that in psoriasis, there's actually a reduction in CLA expression even within the CLA positive compartment. And so from here, we started to first gain insights that there may be a possible defect in homing of these Tregs into the psoriatic skin. And so then when we further interrogated these Treg metaclusters, we found a few differing populations. One of the populations that we found significantly different between psoriasis and healthy controls is this CLA high, C39 low, CCR5 high subset. And so CCR5 is generally known to be an important skin homing receptor. And so what we found is that this subset of CCR5 high Tregs was drastically reduced in psoriasis patients. Not only that, we found that there was also a decrease in the CLA high, Ki67 high, HLA-DR high. So this activated kind of CD4 Treg subset was also significantly reduced within psoriasis. And then furthermore, we also found that there was a CLA high, CXCR3 high subset that was also reduced in psoriasis. And we also know that CXCR3 is an important skin homing receptor. On the contrast of the populations that were increased within psoriasis, we found that there was an increase in the CCR7 high Tregs. And so CCR7 is an important chemokine receptor for migration of T cells into secondary lymphoid organs. And we found that that was significantly increased in psoriasis. And so we wanted to kind of more biasly or do DEG differential protein expression analysis of these Tregs to further kind of interrogate why we think there is a potential reduction or potential impaired function of skin homing of these Tregs. And so what we did is we compared the differential expression of these different proteins on both overall Tregs and within CLA high Tregs. And what we found is that markers in red are markers that are increased in psoriasis, and the markers are in blue are markers that are decreased in psoriasis. And so what we found is that in both Tregs and CLA high Tregs, CCR7 is significantly increased in Treg populations, while in reverse, CCR4 is reduced, CLA is reduced, and CCR5 is also reduced. So all of these skin-relating homing chemokine receptors are all reduced in psoriasis, and on the contrary, CCR7 is increased in psoriasis, and further showing that there might be this potential of an impaired skin homing phenotype of psoriatic Tregs. And so we wanted to kind of validate this a bit more. This is just within circulating Tregs, and we wanted to see like actually within the skin, does this concept hold true? And so we did immunofluorescence of psoriatic skin versus healthy skin. And so what we found similarly is that within the skin, within FOXP3 Tregs, we found that there's an overexpression of CCR7 compared to healthy skin. And so indeed, within psoriasis, almost all of the FOXP3 Tregs will express CCR7 compared to almost none within healthy skin. And we also want to do an in vitro validation of this phenotype. And so we conducted an in vitro transluminal migration assay of these Tregs. And so essentially what we did is we took PBMCs from psoriasis patients and from healthy donors. We then isolated Tregs from those PBMCs, and then we conducted a transluminal migration assay. So we inserted these isolated Tregs into these insert, and then we added the chemoattractant. In this case, we added the CCR7 ligand, CCL19 and CCL21, at the bottom of the well, and we measured the migration of these Tregs into the chemoattractant area. And what we found was that psoriatic Tregs indeed have a significant increase in migration towards CCR7 ligand, CCL19, and CCL21. And so we're thinking that because of the increase in CCR7 expression on these psoriatic Tregs, they're more prone to migrate towards the chemokine ligands, CCL19 and CCL21. And so we also wanted to evaluate this based on patient severity. And so what we did is we we looked at the severity of psoriasis based on the PASI score, and then we tried to correlate this with various markers. And indeed, so right here is a UMAP of those same Tregs, but then divided into mild psoriasis compared to moderate to severe psoriasis. And what we can first see is that CCR4 expression decreases for severity, and also CCR7 expression increases for severity. So similarly, what we can see here is that in the mild group, the severe group, CCR7 is much higher compared to healthy controls. And then actually with the CCR4, we actually see kind of this linear reduction in CCR4 expression that correlates with severity. This is also similar for CXCR3, where we find that the most severe samples from psoriasis patients have the lowest expression of CXCR3, and again, CCR4. And so we're thinking that a big potential reason of these Tregs that need to go into the inflammatory skin and kind of control the ongoing inflammation, because CCR4 and CCR7 is reduced, these Tregs are less prone to go to the skin. And then also, even when they're within the skin, because of this increased CCR7 expression, they're more prone to leave the skin and go circulating to secondary organs. So they're not able to maintain their suppressive function or stay long enough within the skin, the inflammatory skin, to maintain their suppressive function. And so while this is also mostly this is all in circulating PBMCs, we wanted to validate this phenotype also within the skin itself. And so we did a meta-analysis of public single-cell RNA sequencing data sets. And so we aggregated two data sets, the Reynolds data set and the Liu data set that includes both lesional psoriasis skin, non-lesional psoriasis skin, and also healthy skin. And so we combined all that data together. We did integration and then annotation of all the cell subsets within each of these compartments. And what we could find is that, first of all, when we look at Tregs within the skin, when we compare lesional Tregs to non-lesional Tregs, we find actually a very similar finding to our CyTOF circulating findings, where we see that CCR4 expression is reduced in the lesional skin compared to non-lesional. CCR5 expression is also reduced in the lesion compared to non-lesional. And then CCR7 expression is increased in the lesional psoriatic skin compared to non-lesional. Furthermore, when we look at the gene cell-PLG, so this is a gene that encodes CLA, we also see that CLA expression is also reduced within the lesional skin compared to the non-lesional skin, further capitulating upon our findings that we found within circulating Tregs. We also then did ligand receptor interaction analysis, and what we found was that so CCR4, CCL5 interaction, as well as CCR4, CCL22 interaction, was much more significantly enriched in Tregs within the non-lesional skin, whereas these interactions were negligible and not significant in the lesional skins, further indicating that a reduction in the CCR4 expression is kind of leading to much less interactions with the ligands in the skin. And so then going on, we wanted to further integrate this, so we did GSEA GO and KEGG pathway analyses. And so what we found kind of evidently within psoriatic Tregs is that, of course, in psoriasis, we have hallmark pathways of psoriasis, such as TNF-alpha and Th17 signaling being upregulated in psoriasis. TNF is increased here. IL-6 is increased. TNF signaling is also increased, as well as Th17 differentiation. And so these common hallmarks of psoriasis we also found validated in these data sets. But then we also wanted to look at the kind of pairwise, or rather than pairwise, these interactions between all of the ligands and their receptors within Tregs in the skin. And again, we found that CCR7 expression was increased within psoriatic Tregs compared to non-lesional skin. We also see that there's a reduction in CCR4 expression, reduction in CCR5 expression. And then furthermore, this pathway analysis also revealed that there is a reduction in the cell adhesion-related genes such as TMP2, cell PLG, and ETJ. And so all of these genes related to keeping the Tregs within the skin are also reduced within these cirrhotic Tregs. And so in summary of all of these findings, we found that the integrated results of CyTOF and CMSR sequencing data demonstrate that there's an impaired skin tracking property of cirrhotic Tregs. And this, we think, is due to an increase in the secondary lymphoid marker CCR7. And it's also due to a decrease in the skin homing marker CCTLA, CCR4, and CCR5 on cirrhotic Tregs. And so because these cirrhotic Tregs potentially have a reduced propensity to migrate and also persist within the cirrhotic lesion, they are unable to fully exert their immune suppressive function, leading to suboptimal control of the ongoing cirrhotic inflammation. And so at this point, we need to kind of conduct further studies to determine what is the underlying mechanism. Why is CCR7 increased in the first place? Why is CCR4, CCR5, and CLA decreased in the first place? And then that way, we can kind of think about developing therapeutic strategies to promote Treg recruitment and retention within the inflamed tissue. And so with that, I'd like to thank Professor Kim, Professor Hyunjae Kim, and Professor Chungju Park as the corresponding authors leading this study. Also our collaborators at Mount Sinai, Professor Seung-hee Kim Shulse over at the Icahn School of Medicine at Mount Sinai. Also, special thanks to the co-first authors involved in this project, Sang-ah Lim from Nancy University, Yunji Bang from Seoul National University, then also co-authors of this paper, Seung-Jun Kang and Sang-hee Kim. And I'd also like to thank our entire lab for supporting for this project. And then a special thanks to Rick for hosting this seminar, this webinar, and giving me the opportunity to speak today. With that, thank you, and happy to take any questions.